So we have been looking at mineral acquisition by the plants, and I'm updating some of these slides, and I will post the updated slide set probably this afternoon. But this one I did edit and add some things to. So we were looking at last week the idea of cation exchange, which we've gone through, and that idea that you have negatively charged soil particles. The cations can stick to those quite nicely, and then through water flowing across the soil particles, you can imagine where other cations can bump off some other cations that are on the soil particle in an exchange process. And so we've gone through that. The thing I updated on this slide was getting a little bit more about the microbes. And so just pointing out that the presence of the microbes in the soil, just living and doing their thing, can create situations where they help to solubilize the minerals, which means they're able to get the mineral particles off the soil and into the soil water, and that's what the plant needs. And so you may hear this phrased in different ways. I tend to say that they solubilize the minerals because that's kind of the key thing. You need the plant roots have to have access to the minerals in the water solution. They can't just grab it off the soil particle. You may hear it expressed as weathering of rocks, mineral dissolution, rock degradation. Okay, so this is something that microbes are doing. So if you have a healthy soil system in the sense of lots of diversity of microbes in the soil, especially around the roots, then you get this nice action of them releasing these minerals into the soil water. If you don't have these microbes in the soil, then you can see where the plant will start to struggle. And so between these two processes, that's where the bulk of the availability for the roots, the mineral availability is coming from. And so we've worked through the cation exchange. We've looked at that. We've considered the idea that soil pH does play a role in terms of how soluble the minerals are in the water solution of the soil. And so where you see the fatter bars here is where it's more available. So 5.5 to 6.5 for most minerals is the pH range that seems to be the most available. And so let's see. All right, and so the acidic soil system, we talked a little bit about that as well. That's just happening through rainfall and the combination of CO2 mixing in with the water in the soil. And because pH is dependent on the proton concentration, when you have this reaction going on in the soil, you start to see a drop in the pH with more and more protons being released. And so these conditions provide slightly acidic, most soils, slightly acidic, pH 5.5 to 6.5. And that can be very good for mycorrhizal fungi. So fungi like acidic conditions. This is one group of fungi that you want them to be there in the soil. And this is one of my favorite topics to think about. Part of my PhD work was on mycorrhizal fungi. And the first time I learned about mycorrhizal fungi was in a plant phys class when I was an undergrad, and I was just kind of blown away by this idea that you can have two very distinct groups of organisms partnering up in a very intimate association in order to live their lives and have a win-win situation. And up to that point, I've been taking like ecology classes and other classes where they tend to focus on competition. Everything, life is in competition. That's all we do is compete and kill and try to win. And when this was being discussed, that they're actually cooperating with each other and creating a situation where they both are benefiting, I just thought it was wonderful. So I'd like to spend some time talking about this. This is hugely important. It's gaining an interest. I see a lot of businesses popping up where they're offering mycorrhizal fungi for sale, whether you're a home gardener or you are having a big agricultural system. And so this is the idea of you have the fungal partner in the root zone. And so here, this mock-up picture is showing a longitudinal section of the plant root. And on the left, you have the so-called arbuscular endomycorrhizae, where the artist is trying to draw that association, and then the ectomycorrhiza. And so those are the two groups we'll work on. There are other groups of mycorrhizal fungi, certainly, but these are the ones we're going to focus on. You hear this term mycorrhizae. If you hear that term, that is supposed to talk about the actual symbiosis is occurring. You have the plant and fungus hanging out with each other. They're in an established relationship. So that's what that word means. And literally, when you look it up, myco is fungus and rhizae is root. So fungus root. So conservatively, 80% of plants, I've actually seen some scientists push that up to over 90% of plants have mycorrhizal relationships of some sort. And so that makes you think, well, it's probably pretty important if so many plants have established this relationship. And when you think about what this relationship does for both partners, there's some clear benefits. And there's been hundreds, thousands of studies done looking at the benefits of this relationship especially on plant growth and yield of biomass yield. So here's a cute little mock-up trying to show some of the ideas here where you have hyphae, the fungal hyphae, growing off the root. And one thing to appreciate is that fungal hyphae are much more petite in diameter compared to even the finest plant root. So that's a big advantage for the plant, because if the plant roots can't physically access some part of the soil because it's too packed in, too tight, the fungal hyphae can. And so the ability of them to get into tighter nooks and crannies of the soil is a huge win for the plant, rather than the plant trying to evolve these mechanisms where it has ultrafine root. And this picture here, and then the other thing here that they're trying to show is that it's a flow of nutrients back and forth. The plant will give sugar to the fungus so the fungus can stay alive and keep doing its thing. And then in return, the fungus is giving a lot of water that it can get from the soil system, as well as minerals to the plant. And that's not all it does, but that's kind of like the big picture idea of this association. I like this image because it's very visual, very distinct. So the darker lines, that's all root, plant roots. And so you can see the white bar or the white arrow is pointing to, the plant roots. And then you see all this other stuff where they have the arrowhead down here. All of that's all fungal hyphae. So you can see the difference in size, clear difference. And so fungal hyphae are able to get into tighter spaces of the soil compared to the plant roots. So it's a huge win. This table is looking at a couple of different types of mycorrhizal fungi. So I just wanted to point out that even though we're going to cover the top two here, the vesicular or arbuscular mycorrhizae, which is quite a mouthful, or the ectomycorrhizae, then there's also these other types too. Orchids have their own type of mycorrhizae, and ericoid and some mixed monotropoid. So when you look at tables like this that are published, very often the way especially in a table, because you have to limit your number of words in the table, you often see this expression here. It says fungi produce hyphae, arbuscles, and vesicles in the plant root. Okay, so I asterisked that and put a little note down here that they're not in the root cells. That's not how you interpret that, okay? So fungi don't live inside the root cells directly, where they're they're not swimming around in the cytoplasm. That's not what they do. There is always a membrane, a plant-derived membrane that surrounds the fungal hyphae and keeps that barrier and boundary, so it's like the plant is saying, hey, you got to respect my boundaries. There's only so much I'm going to let you in, and then that's it. And so if the fungus accidentally would get into the cytoplasm, actually make physical contact with the cytoplasm, then the plant tends to go, oops, you're a pathogen. That's too intimate. I'm going to kill you. And so it triggers a defense response. And so it depends what source you read, but very often they're kind of vague about that idea. And I just want to make sure that you understand that that is. And here is a paper that was published where they used a electron microscope in order to get at that idea. And they could show that there was always a plant-derived membrane around the fungal hyphae. And so the fungus does have a boundary initiated by the plant to make sure that they're in an association, but it's not reaching into the cytoplasm. And here's another paper that was published. And so when you look at it, if you didn't have the training on electron microscopy, just looking at figure A, you might think, oh, they're just in the cell. They're just kind of there in the cytoplasm, but that's not the case. There's very nice, careful studies have been done where you do have that association of the membrane. This mock-up is trying to show that, where they're trying to show the plant cell membrane is surrounding the fungal hyphae. So I'm got several slides here devoted to this idea because it's a big deal. It's really annoying to me when they published phrases where they make it sound like the fungus is totally swimming around in the cytoplasm. So be careful about that. They are not passing and penetrating the plant cell membranes. Okay. All right. So here's a mock-up of the two main groups I'll focus on. Many crop plants and plants of medicinal interest tend to form associations with the so-called arbuscular mycorrhizal fungi. And then the ectomycorrhizae, on the other hand, they tend to hang out with trees and shrubs. So, and then there's some plants that have both. Some plants can have both types. Some trees in the tropical area along the equatorial line, they can have both ecto and the arbuscular type. So you can see the artist has drawn this where there's some differences between the two. They're trying to show on the right with the ecto, they're trying to show that there's quite a bit of growth of the fungal hyphae right on the surface of the root. And it's quite compacted. It's tissue-like. It's very dense, to the point where if you see a tree that's been like wind blown over and the root systems are exposed, once you know what to look for, you can see the ectomycorrhizal relationship. You'll see it with the naked eye. Hand lens helps, but you can see it with the naked eye. I'll show you some pictures. So that thick growth like that, they call it a mantle. I would like you to know that word, please. So a mantle is just that thick growth of fungal hyphae on the surface of the root. We don't see that with the arbuscular type on the left. They grow on the surface, but it's much thinner, not nearly tissue-like dense growth of a mantle. And then you can see in both cases, the hyphae come off the root. So of course, that's pretty important. You want hyphae coming off the root, penetrating into the soil, into the nooks and crannies that the plant root can't get to, so that they can sop up all that water and the dissolved minerals. Then there's a couple of differences when you look at what's going on in the root tissue. So here you're seeing a bunch of root cells, these mostly parenchyma cells, all next to each other. And then you can see here, there's this Hartig net. Okay, so the Hartig net is, when you look at ectomycorrhizal fungi, what they do is some of their hyphae penetrate into the root tissue and they live in between the cells. So they're in the cell wall space. And so they're right in the cell wall space and they can surround the cells. And so if that's pretty extensive, and if you can imagine all the root cells going away, what you're left with is a net. It looks like a fishing net. And so Dr. Hartig, they named it after him. He was the one who discovered it. And so it's called the Hartig net. And so that's not as intimate as what we see with the arbuscule type of mycorrhizal fungi. Here, what the artist drew is fungal hyphae. It looks like it's in the root cells, but remember, there's always a membrane around that, okay? But you do see that they've gone beyond the cell wall. So what they do, the arbuscule type, they can breach the cell wall and then they kind of hug up to the cell membrane. And they make very close association with the cell membrane. And they can push in a little bit so that the membrane, kind of invaginates a little bit. So that's why the artist drew it like that, where it looks like it's pushed into the cell. So the reason they call them arbuscular type of mycorrhizae, that word means dwarf tree. And if you look, I'll show you some better pictures. The ingrowth here where it looks like there's short little stubby hyphae in that area, that they look like dwarf trees. So that's why it got that mouthful of a name, the arbuscular type. They also form vesicles. So this is fungal hyphae here, where you see that kind of balloon looking structure. And again, it's surrounded by plant cell membrane, but they can breach the cell wall and hug up to the cell membrane of the plant. And they look like balloons or vesicles. And so that's why they're often called vesicular arbuscular mycorrhizal fungi, which abbreviates down to VAM. So sometimes you'll hear me say the word VAM. That's the same thing. Okay, so VAM fungi, I often say that because it's just easier to say than vesicular arbuscular mycorrhizae. And then often we'll refer to the ectomycorrhizae, we just shorten it to ecto. Okay. The other thing too, like around this time of year, if you're out hiking around in the woods here, you may see fruiting bodies or mushrooms right around the base of the trees. Chances are those fruiting bodies are ectomycorrhizal fungi that you're seeing above ground. And so that's just their mode of going through sexual reproduction. So if you see a mushroom, you know the fungi are having sex. And this is their version of getting their sexual spores matured, and then they'll release out into the air currents. Okay, so some similarities, some clear differences. So this is in words what we were just going over on that slide. The thick mantle, okay, the thick mantle around the roots, very tissue-like because it's just so condensed and closely grown together. And then some penetrate between the cortical cells or parenchyma cells. All right, that's cortical is another word for root cell, but they're just parenchyma. Most of them are just parenchyma cells of the root. And so they hanging out in the cell wall space. So that's the Hardig net. And then it's a lot. It's a lot of growth by the fungus. It's a big investment. The fungus does grow pretty luxuriantly. Often the total mass is comparable to the root mass itself. All right, so it's a pretty extensive system. Tend to find the ectomycorrhizae, again, they like to associate with trees and shrubs. So any of the trees around here, shrubs around here, yes, there's definitely ectomycorrhizal fungi hanging out. And you really can't see them unless like I said, if there's a windblown tree, go check out the root system and I'll show you what to look for. And then the fruiting bodies are kind of indirect evidence that you have mycorrhizal fungi, ectomycorrhizal fungi hanging out. So here's clusters of chanterelle, which are edible. Some ectomycorrhizal fungi are edible, and these can taste quite delicious. And then sometimes you see the fairy ring idea where they form a ring around the base of the trees. And again, chances are those are ectomycorrhizal fungi fruiting bodies that are popping up so that they can finish their sexual reproduction and get their spores released. And here you have a conifer seedling that is very young. And this is a nice picture because it's showing the root system is kind of the yellowish, thicker white that you see. And then all of this more ephemeral looking white area here, all of that, that's all fungal hyphae. And so again, a really nice visual showing how the fungal hyphae extend off the root into the soil, really extend out the root system substantially. And with that finer diameter, it's so nice for the plant because now it has access to things it would never have access to because its root system, its roots are too big in diameter for some of that. Here's another nice image. And then sometimes you'll even have these in like the greenhouse, they tend to inoculate conifers that they're growing in the greenhouse before they outplant them to field sites. They tend to inoculate them now. They know that they should inoculate with ectomycorrhizal fungi before they put them out in the field. And then the survival rate of their seedlings increases greatly. Here's another drawing of ectomycorrhizal fungi where they're trying to show that mantle on the surface of the root, the Hartig net as the hyphae go into the root tissue, but just hanging out in the cell wall space. That's where they stop. And here's a cross section of a root where they drew, again, that mantle on the surface. You can see they're trying to show how thick it is. It's very tissue-like. And then the Hartig net, this gives a little bit better perspective of the net idea of the fungal hyphal growth. And here's a dead giveaway. So if you go out hiking and you see an overturned root system, look for this. This is what you should look for. Those Y-shaped finer roots. And so a couple of things happen here. As the fungus is approaching the plant root, they're communicating with each other. It's like they're having a conversation through chemicals where they're saying, hey, I'm a friend to you. Let's get together and form this relationship. And then the plant will go, yeah, I agree. I think this could be a win-win. And so then they will start forming this relationship. One of the first things that happens is the chemicals released by the fungus get the finer roots to form a Y shape. So that's the root actually growing into a Y shape. They think to increase surface area. And then the fungus starts colonizing that, growing that thick mantle around that. And so you often see that pattern. I have some real images in a little bit here that I'll show you. And you can see that with the naked eye. They're small, but if you see a Y shaped, especially on the conifer trees around here, that's pretty standard. I mean, it's like, okay, yep, there's mycorrhizal fungi, ectomycorrhizal fungi. So normally plant roots and finer plant roots just grow straight. So if you see that Y-shaped growth, that's kind of a dead giveaway that you got the ectomycorrhizal fungi. And then they do look thicker. You can train your eye after a bit, you can see that they are thicker. Here's a really nice image showing how thick the mantle can get around the root. So this is a root and cross section, and all that blue-green, that's all fungal hyphae, just really grown, quite compressed onto the surface of the root. And then you can see also the blue-green hyphae coming into the root tissue and growing around the root cells. So a very nice image. Here's another image where it's not quite so bright, but you can still see the fungal hyphae, the Hartig net around the cells, and then part of the mantle. Here's a very nice difference. The roots on the left were inoculated. The soil system was inoculated with ectomycorrhizal fungi compared to the ones on the right. And so I think you can clearly see how much thicker the roots are on the left. And if you look closely, it's a little grainy, but you can see the Y shape on some of them. So here's a nice image showing that Y-shaped growth pattern by the roots. And in this case, the fungal hyphae are a different color than the roots. So you can actually pick that up quite easily. And you can see how they're thicker compared to some of the other roots there that aren't covered by the mycorrhizal fungus. And then when you process roots and look at them under the microscope, very often you destroy the fine hyphae that are growing off the surface. So just don't forget, you have plenty of hyphae coming off the surface of the mantle, growing out into the soil. And so sometimes if you carefully excavate around the roots, you can see the fungal hyphae in the soil, that white webbing look. You may have noticed that at some point. That's all probably mycorrhizal fungi by the roots. if that's where you're digging around. And then this image is what you would see with the naked eye. So the top left image is under a compound microscope. The bottom right is just with the naked eye, what you would see. So you can see that. I think, if you look closely, you can see some of the branched finer roots, and they're thickened up. So I worked with ectomycorrhizal fungi when I was a grad student because back then we knew how to grow them in the lab, separate from the plant host. And so they were easier to work with compared to the vesicular or arbuscular mycorrhizae, that they didn't know how to grow that in the lab yet. And so my research focused on a couple of different kinds of mycorrhizal fungi that hang out with red pine trees, because they used to be called Mead Paper Company, not the Mead Paper Company anymore, but they had plantation sites here in the UP where they wanted red pine trees growing for paper production. And so we were able to use some of their field plots for our research. So here, different fungi, ectomycorrhizal fungi, you can see the pigmentation differences. Again, some of the nice branching, that Y-shaped branching, and how it's very thick compared to a non-covered root. And some other images here too. So once you train your eye to this, it's not too hard to find if you're out hiking around. You can see them. The bottom right image again is pretty much with the naked eye. And this one was very carefully excavated where you can see fungal hyphae, and then the really cool looking Y-shaped root systems. They're heavily colonized by the fungus. They're so cute. So we have both internal hyphae going into the root tissue to help with transfer of nutrients to the plant host. You have water getting transferred, minerals getting transferred, other things getting transferred, which we'll talk about a little bit. But that's all coming from the soil system. You have that external hyphae going off the fungal mantle, going out into the soil and able to get access to all those nutrients and chemicals that the plant needs. And in return, the plant is feeding the fungus with sugar. And depending on the plant species, it can be substantial. Like the plant really gives up a lot of its sugar to the fungus. Some reports on, I think it was Douglas fir out west in Vancouver area, in that area, British Columbia, there we go. And they did some radioactive tracing of the carbon to the fungus, and they showed like 30% of the carbon the plant made went to the fungus. So that's a substantial input of resources, and really makes you think that the plant must consider this pretty important to maintain that level of feeding of its resources to the fungus. So a type of ectomycorrhizal fungi, Lacaria bicolor. This was one that I worked on for my grad work. This one I chose because at that time it was being heavily studied by people who were into mycorrhizal fungi, and they started sequencing its genome. And so I wanted to work with it since they knew more about this fungus than any other type of mycorrhizal fungi. The other reason I chose it is that it's a very common mycorrhizal fungus. It forms relationships with a lot of different, primarily pine trees, conifers, but it's very common. It has a broad host range. And so it's pretty common that you'll find it in a variety of coniferous forests around the world. Its fruiting body is, they call it bicolor because it can be tan and purple. So it's two colors. And you can kind of see that where the purpley base of the stem, sometimes under the cap, it looks purple, and then the rest of it looks tannish. When you grow it on an auger plate, it does that bicolor look again. The young vigorous growth is purple. So when you grow a fungus on a plate, by the way, if you've ever looked at that, what they did here is they put a chunk of fungal hyphae in the center of a Petri plate, and then they incubated it. And fungi grow out from the center like that, pretty much in a radial pattern. So it forms a circle of growth like that. And anytime, like if I wanted to take this and subculture, like, oh, I want to make more of these plates, I want to grow it up some more. What I would do is take the very edge, which is the purpley part. So the very edge of the hyphae, that's the most vigorous and young portion of this culture. The tannish part is pretty much dying off, and it's the purple part that you want if you want to grab some really healthy tissue and fungal hyphae and get it transferred. So you can see this image, it's growing on a Petri plate. It's easy to just have it growing without its host. You don't have to have the plant there to get it to grow. So they figured out ways to get this fungus to be happy in the lab and grow on its own. And then they were able to do a lot of the more specific experiments that they wanted to do with Lacaria. Just some images of the fruiting body, especially that lower one, you can really see the purple. So we have it around here. It's definitely around here. And it looks like that under the microscope when it forms with the roots, the ectomycorrhizal relationship. So they've done all kinds of research on it. They've looked at how these are Petri plate experiments where they put little seedlings going with their roots growing, and then they inoculate the Lacaria bicolor onto the plate. And what they're doing here is they're assessing how this relationship forms. How do they decide that they want to partner up? And there's a lot of chemical communication going on, making sure that they're both on the same page, so to speak, and they're willing to partner up and form this relationship. And so they've done a lot of these studies, and they have sequenced Lacaria bicolor, its whole genome. One of the experiments that they were doing was surprising though. This was can't remember where this was published. It was just like a normal magazine. It wasn't a scientific paper. Root fungi can turn pine trees into carnivores, or at least accomplices. And so root fungi may confer dark but useful powers on their plant hosts. So what they were running a different experiment and kind of stumbled on this issue where, in the soil, you have these springtail insects and the mites. And they were looking at this feeding experiment, and they saw that the springtails, less than 5% of the springtails survived the fungus presence there. And it was Lacaria bicolor. And so they said, incriminatingly, all the dead springtails had fungus fibers riddling their little corpses. Instead of springtails eating the fungus, the reverse happened. So the table got turned. The fungus, instead of being eaten, started to eat the insects. And so they went, what? That's not supposed to happen. And so what they were discovering is insects, their exoskeleton has a lot of nitrogen. That's a really rich source of nitrogen. So 25% of the nitrogen showing up in the plant tissue that the fungus was hanging out with was from the springtails. So the fungus was attacking the springtails, killing them, eating them, taking up their nitrogen and giving it to the tree. And they could track that flow of nitrogen from the dead insect to the fungus to the tree. And so the plants grew better when fertilized by the hapless springtails. Weighing more, the plants weighed more at the end of the experiment than their control counterparts. So this was something that they did not expect. Less than 10% of the springtails at the end of the experiment were alive. An average of just 47 wily survivors out of 500 that they started with. So it was an entertaining read, but basically this is kind of a serendipitous, what's going on here thing, where they started to realize that the mycorrhizal fungi have more aggressive ways to get access to nitrogen. So you can imagine if this plant and this fungus are under a situation where their nitrogen levels are dropping in the soil, that the fungus could turn their attention to the insects and start attacking the insects. And fungi are great at killing insects, actually. I just purchased Bovaria bassiana, which is a fungus that we're going to spray out in the greenhouse because we were having some insect infestations going on. And the fungus, once it makes contact with the insect, it just starts growing over the insect and sending out enzymes to kill and digest the insects. And so fungi are just really good at that. And so they were surprised to see that the mycorrhizal fungi are able to get access to nitrogen, just regular nitrogen in the soil, but also nitrogen locked up in the exoskeleton. And so that's a nice versatile way for the fungus to get access to nitrogen. Here's another ectomycorrhizal fungi that I worked with, Xenococcum geophyllum. And this one also, they were able to figure out how to get it to grow in the lab. And so it was fairly easy to work with as a grad student. It is super widespread. It's found all over the world. And it, again, broad host range. It can hang out with all kinds of different plants. And it's able to get up into the Arctic and even subtropical areas. So lots of versatility in what kinds of habitats it prefers. The downside is it grows slowly in the lab, but if you have some patience, it forms this lovely black colony on the surface of the plate. You just have to take your time with it. But this one was interesting too because it can secrete antibiotics, and so mycorrhizal fungi are known to like actively help the plant defend itself by excreting out antibiotics to kill off plant pathogens. And so I was very excited about that because I wanted to do a lot of work with biocontrol agents. And so I thought this would be a great thing to pursue, looking at this fungus and its ability to help defend the plant. But there's all kinds of others, ectomycorrhizal fungi. There's many, many species. Just depends what plant you're looking at. Oh, and like one tree can have multiple ectomycorrhizal species. It doesn't have to just form with Xenococcum or just with Leucaria. You can have a bunch. In fact, when I was a grad student, a fellow grad student up in the forestry department, that was his thesis. He was looking at ectomycorrhizal succession on trees. And so he could show that when you put a red pine seedling in the soil, you outplant it, he was able to show what mycorrhizal fungi came in first and colonized the roots. And then over time, he sampled every few months, and he did this project for 5 years, and he could show this succession of mycorrhizal fungi over time. And so we know that that's going on underground as well. And you can have multiple species like by the time that tree is mature and really cooking along, he could show multiple different species of mycorrhizal fungi on the roots. So lots of stuff going on underground. I'll mention a little bit more about mycorrhizal fungi in general when we start looking at some key nutrients that the plant needs, because they're heavily involved in getting access to nutrients that the plant has a really tough time getting access to. So I'm going to come back to mycorrhizal fungi and the ectos, but let me turn my attention to the VAM, so that's the vesicular arbuscular mycorrhizal fungi, and it's easy to say VAM. Now I see in the literature, they've dropped off the V, and so you'll just see AM for arbuscular mycorrhizae. I don't know why they're dropping the V off, because they still form vesicles, but it's not a very good acronym, AM. If I say AM to you, that doesn't, VAM is a little bit more distinctive, so I'm going to stick with VAM, but just be aware that the literature is dropping the V off. I don't know. Anyways, these don't have that thick mantle like we were just seeing. They do grow on the surface, the hyphae does, but it's very wafty and ephemeral. And but they still extend their hyphae off the root, way out into the soil. They still have hyphae going into the root tissue. And just remember, they're not penetrating the cell membrane. They're not doing that. The hyphae aren't swimming around in direct contact with the cytoplasm. They form those vesicles and arbuscles. I'll show you some pictures. These, when you look at their biomass, because they don't form that thick mantle, their biomass isn't very good, relatively. So it's only like 10% of the root weight. But that doesn't mean they're not useful. They're hugely useful to the plants. And so they just don't grow as robustly on the roots. So most of the flowering plants, the so-called herbaceous angiosperms that don't form woody material in their stems in particular. So, tomato, potato, cannabis, mint, basil, I mean, the list goes on and on and on and on. So you will have these types of mycorrhizal fungi hanging out with them. So again, just showing a visual that you have not much growth on the surface. You certainly have growth coming off the root, and then these vesicles and arbuscles that can get past the cell wall and bump up to the cell membrane. And they call them arbuscles because that's a really nice image. Doesn't that look like a little shrub or dwarf tree? And so that's what the word arbuscle means. And so this is an electron microscope picture of a root that's been stained well enough where it does look like, that's all fungal hyphae, but it looks like dwarf trees. And then here they took a longitudinal section of a root and did a staining technique for fungal hyphae. And you can see the roots look, like the brick pattern. And then the fungal hyphae is the darker, grainier looking things. So most of these are arbuscles, the dwarf tree idea. Here's a better image, I think. So there's been students in this class for the research project. They want to look at that. One of their treatments will be mycorrhizal fungi. And then we asked them to look at them under the microscope and do some staining. And it's a pretty simple staining procedure that you can use to actually verify that you did get the relationship. Because just because you put the mycorrhizal fungi into the soil near the plant, it's not a guarantee they're going to form a relationship. They have to do a lot of talking to each other and decide, so to speak, that they want to do this. And the way the plant might decide not to do it would be, and I had this problem with one of my earlier grad students. She was working with ectomycorrhizal fungi. She put the fungi into the soil, had her red pine seeds going, and they never formed a relationship. And we're like, what is going on here? We have perfect conditions for both fungus and plant. What is going on? But they would not form a relationship. They lived separate lives. They were alive in the soil, but they wouldn't form the relationship. Well, if you were a plant and you are getting all the light you want, you're getting fed with a hydroponic solution, so you're getting all the minerals you want, all the water you want, would you want to form a partnership where you might have to give up 30% of your resources? I'm fine by myself. I got all the resources being fed to me. I'm even being kept free of pests. So if you have a sweet life, which is what we did, we accidentally gave our plants too sweet of a life. And they're like, I see you there, but I don't want a partnership. I'm doing fine. So the way we had to get around that is stress the plant. We had to say, no, we're going to stress you a little bit. So you decide to partner up and join forces. And the easiest way to do that is to limit phosphorus. If you take phosphorus content down in the soil, then the plant goes, ooh, I need a partner. And they will start to form the relationship. So just because you inoculate into the soil doesn't guarantee that you're going to get a relationship. And so this staining technique is a nice way to verify that it did actually happen. Okay, so we have many species of plants that are reliant on the VAM type fungi. And so you can, like I said, there's one thing about VAM fungi, by the way, they are very challenging to grow in the lab to do research with them because we don't really know a solid way to grow them separate from the plant host. They're so dependent on their plant host that we can't figure out how to grow them in the lab all by themselves. So when you buy VAM fungi from a company, it'll say something on the package like, 80% of by weight of VAM propagules and plant matter or something like that. And so basically you're getting a mix of a little bit of the plant host along with the fungal hyphae and spores. And then they tell you to sprinkle that into your soil system. So clear advantages, if so many plants are doing this, there must be a clear advantage to the plant. And yes, so many studies over and over and over have shown that the plant gets increased access to P and N and K. I've ranked this in order. I did that on purpose. Phosphorus is a big one. Plants have a really hard time getting access to phosphorus, and I'll explain a little bit more about that later. Mycorrhizal fungi are awesome at it. They can get access to really precipitated out phosphorus, and then they'll give it to the plant. So that's a huge win for the plant. But also nitrogen and potassium. Those three, which are, you know, the three big ones for the plant to those are all macronutrients. Water, of course, because the fungal hyphae can get into all types of narrow spaces in the soil and get that extra water. And there's just great stimulation of seedlings. If you inoculate your seedlings with mycorrhizal fungi, they get off to a really solid start. They can be protected physically. That mantle around the root is very physically protective against creepy crawlies that might be coming along, or other microbes trying to kill them off with chemicals. We also know mycorrhizal fungi can actively secrete chemicals to help defend the plant. So you have that going on as well. So many plant species have been studied over and over. It's clear that this is a win for the plant. And the fungus also, as long as the plant keeps feeding the fungus wins in this relationship too. So Great White is a company that I've purchased the VAM fungi from many times and have had good success with them. And so these are just some of their images showing the without pictures of the mycorrhizal fungi. Yes. Yep, I use it in my garden. There's some arguments. Some people say, well, there's mycorrhizal fungi already in your garden. Well, not my garden soil. I started with poor garden soil that I've had to amend over time. So this does make a difference for sure. And so now, there's a lot of hydroponic growth of plants, right? And so I have a master's student right now who's trying to finish up her thesis writing and all of that. And she did a study looking at adding in mycorrhizal fungi, and we picked certain beneficial bacteria that she grew up in the lab and spiked that into the hydroponic system to see what would happen. She worked with spinach, and then she compared that to soil growth, doing the same treatments into soil, and was looking at persistence of the microbes. Was there any disease pressure, especially in hydroponic systems? Did it cause any issues? And then what did it do to the plant in terms of biomass, nutrient content? And so I think there's a lot of potential for using mycorrhizal fungi and beneficial bacteria in hydroponic systems. I think the plant would actually do better and produce better quality nutrients. If you're going for edible or even the medicinal chemicals can also be increased if you get a system of application that works. So lots of potential there. I'm hoping she'll be finding some interesting results from that. Okay, so this idea that you have these mycorrhizal fungi and beneficial bacteria in the soil system, all playing their role for helping the plant get access to nutrients. I'll explain this image again. It's too late now, we're over time. So I'll get into this a little bit on Wednesday. We'll start there.